Cold, maybe disease, maybe the flu. These are all things that I think a lot of people associate with viruses, things that make you sick. And along those lines, when you look at these words, and these are just some of the words that I came up with when I was brainstorming, is that almost all of them are bad or have sort of a negative connotation. And that's because, like the cold or the flu, viruses often cause disease, they cause death, they cause suffering. But that's not the whole story, and that's what we are here to tell you today. So a quick outline of today's lecture. I'm going to start with a quick introduction to viruses and how they work. And then I'm going to talk about how viruses interact with our human genome, with the DNA inside of us. And Katie, in the second half, is going to talk about the viruses that are inside of our body. And throughout the talk, there will be some words that are bolded and highlighted in green. Those are the words that are on the handout that you might have picked up if you want to reference them and look up their definitions. All right, so a quick introduction to viruses. First of all, what is a virus? A virus is a very, very small infectious agent that only replicates inside the cells of other organisms. So it's this kind of unique organism where it can't really live on its own. It has a particle, and the particle can survive on the desk, anywhere in the world for a short amount of time, but it can't reproduce. It can't propagate itself without being inside another organism. And that can be a human, that can be an animal, that can be a plant, it can even be a bacteria. And because these viruses can take a lot of different forms, but they have sort of a general structure. So here are 3 examples of what a virus might look like if you looked at it. And even though they look different, they all sort of have the same structure, which is that they have a genetic material, which is inside of them. They have DNA, which what we also have and all animals and plants have. But viruses can also have RNA, which is just a variation on that. And I'm going to talk a little bit more about the relationship between those two later. And viruses also have a capsid, sort of a protein coat that encloses their DNA or their RNA. And some viruses have what we call a viral envelope that surrounds the capsid, and the viral envelope is often what interacts with the host cell. And sometimes these proteins that are sticking out on the top are what are used to actually get in the cell and infect it. And so you'll see that this other virus that I drew on the left also has these proteins on the outside, even though they look a little bit different. And there are many types of viruses, many of which you've actually heard of. There are lots of diseases that you might not think of as viruses, but are actually viruses. So here are just some that I thought of. Influenza is the one that I think, unfortunately, we're most familiar with at this type of year. It's a virus, it's an RNA virus. Then we have viruses like smallpox, which are fortunately not around, but they are viruses. They are DNA viruses, so they just have a slightly different type of genetic material inside of them, but they still have the same basic structure. And then there's also HIV, Ebola, many different viruses, all which again have this sort of genetic material inside, surrounded by a protein coat. To give you a sense of scale, I'm going to talk a little bit about what the different cells are inside the human body. So if we look at a human cell, just to give you a reference point, human cells are about 10 times smaller than the width of a human hair. So if you pull out a hair and imagine what 10 times smaller than that is, they're pretty small. And around these human cells, all throughout our body, we also have bacteria. And bacteria are even smaller than that. They're usually about 5 times smaller than a human cell. And then going down one level further, we also have viruses in our body. And those are even smaller, usually 40 to 100 times smaller than a bacterial cell. So as a result, viruses are very simple. They tend to be just the genetic material surrounded by this coat, and that's it. And the interesting thing about this is that even though viruses are really simple, they have this DNA inside of them. And, as I wanted to mention, only 10% of the cells in our body are human. So I just pointed out all the bacteria in here, and this I just wanted to make this point because this is what Katie will be talking about later. So as I started saying, viruses have this genetic material, and human cells also have this genetic material, and bacteria also have this genetic material. But the fascinating thing that I want to highlight today is that if you look closely at the genetic material in our own bodies, in our DNA, you find that some of our DNA actually actually matches what we see in viruses. So there's some viral DNA, some genes that look like virus genes that are inside our own genome. And the even more surprising fact is that almost 10% of our DNA is thought to be viral, is thought to be made up of pieces of viruses that got in there. So I'm going to tell you about how they got there and what they're doing there. But before we understand that, we need to understand how viruses work and what the viral life cycle looks like. And that starts with what we like to call the central dogma of biology. And this is the idea that the DNA in our cells is something like the blueprint. It's what everything in our body is made out of. And inside of our bodies, this DNA is copied, and RNA, which is a similar genetic material, is made out of this DNA. And this RNA is actually what is read by certain proteins in our body to make other proteins, which actually do all the functions inside of our cells and inside of our bodies. They're responsible for everything working, both in us and other organisms. And the reason we call this the central dogma is because all walks of life have this sort of pattern. They start from DNA, the blueprint of everything, that is used to make RNA, and that is used to make protein. One side note is that some viruses actually start at the RNA step. They take out this DNA just because it's simpler. But the same principle applies. The RNA is used to make protein. So let's use this concept to understand the basic viral life cycle. And I'm going to use flu as an example. It's a simple virus. It's an RNA virus. Just easier to draw. Don't have to draw the DNA step. So you can see that this flu virus up here is going to infect a human cell. And when the flu virus infects the human cell, it's going to release its RNA, its genetic material, into the cell. And then this genetic material is going to move into the nucleus of the cell, which is where the DNA of the human. And that's an important step because, as I mentioned, viruses can't exist outside cells. They need cells to reproduce. So what the virus is actually going to do is sort of co-opt the machinery that's inside the human cell and use human cell machinery to copy itself. So what I'm showing here is that now the virus has used what's inside the human cell to take its own RNA and make protein. And so it's made the viral protein. So what I've mentioned, these proteins on the outside in yellow and green, this capsid protein. So it used the machinery inside the cell to turn its RNA into protein and also to copy its own RNA. So now you have all the parts that you need to make a new virus. So this new RNA, that's a copy of the original virus RNA, and the new proteins can come together, make new virus, often killing the host cell in the process. And this new virus can go on to infect other cells, either in the same organism or if it's released into another organism. So that's sort of the general viral life cycle. Before I move on to talk about how these viruses got into our DNA, I just want to pause for questions and see if anyone has any questions about how viruses work or how they get inside cells. Yeah?
Can viruses be said to eat?
Do they said to eat? I don't think so. I think viruses just tend to go inside cells and replicate. But there's this sort of interesting idea where like viruses aren't alive in the same sense that many other organisms are alive, and that's actually a sort of interesting debate. So they don't perform a lot of the functions that we think of as like living beings, like eating. Yeah.
So I saw viruses like enter the membrane and then leave some proteins behind, and then do they actually go into the nucleus of the cell?
Mm-hmm. Yeah, so the question was whether the viruses go into the nucleus of the cell. And yeah, so they actually get into the nucleus, and that's where they can take all the machinery, because the DNA of, like, for example, humans is copied in the nucleus, so they need the proteins that are there specifically to copy their RNA. Other questions? Yeah.
What causes the cell to eventually burst? Like, what is the actual mechanism that bursts?
Yeah, the question was what causes the cell to burst. And it's actually interesting that not all cells physically burst. It's sort of a common misconception in virology that the cell physically explodes. It's possible that it does, but a lot of times there are a few different mechanisms whereby the cell is just overwhelmed, or it doesn't have the capacity to copy its own DNA anymore. So perhaps it doesn't burst, but it can't copy itself. So once it dies, it can't be passed on. Yeah.
So if viruses don't eat, then are they able to take any energy from their environment, or do they have all the energy they're going to have when they're created and then just run out?
So the question was whether viruses take energy from the environment or whether they have it all in them to start with. That's an interesting question. We don't really talk about energy too much with viruses. I think it's because they don't have these normal processes of they don't have to eat to survive. All they really need is, there isn't much to them. It's just the DNA, and all a virus really is this DNA or RNA, and it gets copied and transported. So I think if you want to think of it in terms of energy, it's really using the energy of the cell. It's not providing any of its own machinery to copy and reproduce. Yeah, some people call it sort of a parasitic relationship. It's sort of passive. It's not actively reproducing. It needs to rely on some other organism's energy to do so. Oh.
When you say it only lives within a cell, you kind of mean it's only doing something when it's within a cell?
Yeah, the question was, when I say viruses only live within a cell, as if they're only doing something within a cell. Yeah, so the virus particles that I showed you, those can exist outside of cells. They can, when I sneeze, they can just sort of land on this table and sit there, but they can't really move on their own. They can't reproduce on their own. They rely on being able to insert in the cell. The one function they do is they are, attached to cells and get inside of them, but they don't really serve any other function otherwise. Yeah, up there.
So if viruses aren't really alive, how do you stop them from being active and replicating? So when someone, when they eradicated smallpox, how do you stop it from being an active virus?
And the question is, how do we stop viruses from being active and from infecting us? That's a great question. Our immune systems tend to be pretty good at that. And Katie's going to talk a little bit about how our immune system works in the second half of the talk. But the idea is that we have mechanisms inside our body that fight off foreign particles. And one of those particles is sometimes a virus. Yeah, one more.
What does the cell do to go into, what does the virus do to get into the cell that it's host?
Oh, yeah. So the question is, what does a virus do to actually get into its host cell? So you saw a lot of those viruses have those proteins that stick out from the outside, and many times those proteins have recognize a certain protein on the outside of a cell. So they sort of make a match, or they fit. So when they come in contact with the right kind of cell, they can just sort of push into it and enter it. It's an actually really interesting case of evolution where the proteins on the viruses sort of match the proteins of certain organisms. And if the organism changes that protein, then the virus can no longer get in, so then the virus has to change. All right, hopefully we all have a sort of good understanding of how viruses work. I'm going to move on because I think this retrovirus part is really cool, and you'll learn what that means in just a second. So I'm going to talk about viruses in the human genome. As I mentioned, how we got viruses in our DNA. And more specifically, there are certain types of viruses that can actually insert their genetic material into the DNA, not just the cells, the actual DNA of other organisms, including humans. So what I'm showing here is maybe this black DNA is our human DNA, and the pink is the virus that has inserted itself in there. And this is a special type of virus that's called a retrovirus. So the big question is, how did it get in? And so remember I was telling you about the central dogma of biology. Everything works this way, where we have DNA that's converted into RNA by making a copy. Well, retroviruses are kind of special in that they can actually do the opposite. They can go backwards. They can take RNA, so there can be an RNA virus, and they can convert that into DNA. using a special enzyme that only retroviruses have called reverse transcriptase. So it's reverse, it's backwards. And then they have another enzyme where they can take their DNA and actually stick it inside the DNA of another organism. So now there's DNA of the virus in the human DNA. So when the human DNA gets copied, which is a normal process how our cells replicate, the little bit of virus in there gets copied too. And so actually the human cell is inadvertently making a new virus every time it copies itself. So these are really interesting viruses that have found a new way to make sure they get replicated inside of the cell of their host. And so if we're looking at the life cycle again, here I'm drawing an example of a retrovirus, one of these viruses that can insert itself. HIV is the most commonly heard of retrovirus that can infect humans. And so similarly, the HIV virus, the particle, can attach itself to a host cell, in this case a human cell, and it's going to release its RNA, this is also an RNA virus, into the cell, which is going to move into the nucleus just as before. But now there's an extra step. The RNA of the virus is going to get turned into DNA using that special enzyme that the virus has called reverse transcriptase. Now it's going to integrate itself into the host DNA. And then down here, when the host DNA copies itself as part of its normal life cycle, it's going to inadvertently make virus RNA also, and also make all the proteins needed from this RNA to make a new virus. So now the virus can, again, make new viruses, that can go on to infect other cells. So it's a small variation in the life cycle that actually has really interesting implications for how the virus survives. Okay, but if what I said is true, that 10% of our genome is actually viral, why aren't we sick all the time? Why don't we have viruses like all over inside us making us sick? And the answer is that, well, not all retroviruses make us sick, and that's because we can classify them as sort of two types. And the first type is what I just showed you in the HIV example. These are viruses that are infectious, that can go after cells and get into them, and that can be passed on between people. So HIV is one example where you can get HIV from another person. If you come in contact with that person and they transfer a viral particle to you, you're going to get sick. But there's another way that we can think of retroviruses, and these are called endogenous retroviruses, and these are inside our DNA already. And these ones aren't passed on. There are no actual particles produced, and this is sort of sometimes a hard concept to grasp, where it's not really a virus that I'm talking about. There's no viral particle being made. Here I'm just talking about the piece of viral DNA that's inside the human DNA, and that's what I'm calling an endogenous retrovirus. And these ones can be passed on because they're integrated in my DNA, and when I have offspring, my offspring are going to get that DNA as well, which contains the virus part. And the reason why that virus part isn't producing new virus is because mutations just sort of happen all the time. And if there's a mutation that happens in the course of the virus being passed down through generations, if a mutation happens in the virus, then it might not work anymore, and you don't have the viral protein, so you can't make a new virus. And just as a reminder, when I'm talking about a mutation, if we zoom into this DNA here, it's actually a series of letters, or what we've labeled as letters. And a mutation is sometimes as small as one letter, one base pair being off. And if this is in the wrong place in the virus, then the viral protein might not work, and we can't make a new virus, and it can't make us sick. Which leaves us with all of these pieces of viruses in our genome, these fragments that are not whole, they're not correct or complete enough to create viral particles that can infect us, but they sort of just sit there in the genome, perhaps doing nothing at all. And there's actually a term that we use for this. We call this sort of junk DNA. It's this part of DNA inside our own cells that we don't know what it's doing. And a lot of it comes from viruses. It's not harming us, it's not hurting us, it's just sort of there as a remnant or a record of the viruses that we've interacted with in the past. So that was a lot, so I'm just going to summarize and then pause for questions once more, and then after questions, I'm going to tell you about how we can actually use retroviruses for good, not just bad. So before I move into questions, just a quick summary. The viruses that can insert themselves are called retroviruses. They go backwards. They convert RNA to DNA, which is sort of the backwards of the central dogma. And the crazy thing is that 10% of the DNA in our body is actually viral. It comes from viruses at some point. So, any questions? All right, I'll start over here.
Because you have mutations that would make some endogenous viruses active.
Like if it wasn't active before.
Right.
Yes.
Everything happens all the time, so.
Yeah, so the question is, if there's an endogenous retrovirus, could you have a mutation that makes it active again? And I would say that it's possible, it's just really unlikely, just because there are so many places in our genome that a mutation happens, and so usually what happens is the mutation hits the virus like once in many, many years of being passed on, and that's what makes the virus endogenous. And the chance that the mutation is going to reverse before another mutation happens is just a really small chance. So I guess it's possible. It's just one of those things that tends not to happen. All right, let's go here.
We have bacteria that are beneficial in our bodies.
Mm-hmm.
Are there also viruses that are beneficial in our body?
Just wait.
Yeah, that's the answer. So the question was whether there are viruses that are beneficial, and Katie jumped in because this is what she's going to talk about in the second half. Yeah?
Were the endogenous viruses once harmful
Mm-hmm.
to us? And then they just weren't harmful enough to kill us, so we...
Yeah, so the question is, were the endogenous viruses once harmful? And so yeah, all viruses start out as these exogenous, they're outside, they infect. And what happens is if you infect a certain type of cell, a germ cell like an egg or a sperm that can be passed on, then you can pass on this, and what happens is usually it will only get passed on if the person isn't killed, and so over time the mutations accumulate and it becomes not infectious anymore. Yes, trying to pick new people.
Is there a mechanism that targets the viral DNA to a specific region in the host DNA? Imagine there would have to be if it's been used in gene delivery.
Yeah, so the question is whether the virus, when it gets into the host DNA, targets a specific region. And that's a great question because that's something we're trying to work out. It's definitely true that some viruses target certain types of cells, and they like to get into certain types of cells. And what I'll talk about in the next part is how we're trying to figure out how specific we can be at targeting, and that's something that we're not perfect at yet. The virus can sort of integrate anywhere or in many different places. Now your question.
Does this kind of relationship between the viral genome and the human genome affect the generations of a miasm?
The generations of what?
Of a miasm of diseases through generations.
So
Humans.
yeah, so you're asking is whether the integration of the virus affect additional diseases or?
Let's say the progeny of someone who had tuberculosis.
Mm-hmm.
That would affect the DNA of the person that's, the offspring. That there's some kind of genetic record
Yeah.
that creates a vulnerability to a specific, virus or...
Yeah, so the question is if the record, if the viral DNA inside of us actually affects if we get sick again. Is that the question?
No,
Oh.
I mean the progeny, the constitution of the next.
Oh, okay, yeah. So when the retrovirus actually gets in and gets passed on, it stays in all the progeny. And so all the progeny will have that piece of virus. And so that's why sometimes people consider retroviruses in our genome as a record exactly of all of the viruses our ancestors have ever interacted with. The important thing to note is that it's a pretty rare event for a virus to actually integrate and get passed on. So it's not just any virus or any infectious virus that can get in. It has to be a retrovirus, and it has to it doesn't happen all the time. It's not like HIV isn't being passed on in that way, even though it's a retrovirus. Does that answer your question?
Well, I mean, there's babies born with AIDS.
Yeah, so babies are born with AIDS, but that's actually because they got a virus particle from the blood of their mother, not because it was in their DNA. Yeah, that's a very confusing point. It's worth mentioning that there are very few viral integrations if you think about the course of history. We have viral integrations over the course of 30 million years, and we think that the last one was 30,000 years ago. So it doesn't happen every single day. Other question? Yeah, up there.
Why does an endogenous virus take the time to convert its RNA into DNA? Why can't it just multiply itself as an RNA strand already in the cell, in the host cell?
Yeah, that's a great question. So the question is, why does the retrovirus take time to convert into DNA? Why doesn't it just live like a normal virus? And I don't know if I know the exact answer to this question, but my guess is that if you can get into the DNA of your host and a mutation doesn't happen, then you're guaranteed to be replicated forever. You don't have to go find a new cell, you're just built into the host DNA. So I'm sure there are examples of, maybe not a human, but an animal or a plant host where the virus is just always being made and it's always surviving. So it's very advantageous if you can do that.
So actually, I'll chime in there. The RNA is actually very unstable inside the cell. It needs to be, it actually gets quickly degraded. And so in order to preserve the integrity, the virus will integrate into DNA and harness the cells into machinery. RNA doesn't last very long.
Yeah. Yeah.
Are there theories as to how regular the accumulation of retroviruses throughout history was?
Yes, there are. So the question is whether there are theories about how regular the accumulation of retroviruses is throughout history. And there are some interesting theories talking about as humans evolved from monkeys and apes and how that might have actually slowed down. And I personally am not exactly sure why, and I'm not sure if researchers know why, but that's definitely a field of study, is trying to understand what that rate was, and if we can use that to track changes in our own evolution or something like that. Yes?
What was the timescale for the life cycle that you showed us? From getting into a cell to reproducing and potentially killing the cell?
Yeah, so the question is, how fast is the life cycle of the virus? And viruses, there's definitely a range. It depends on the specific virus and how big it is. But viruses replicate generally pretty fast in terms of compared to other organisms. So I think we're talking on the order of minutes or hours, not more than days or weeks. It's a pretty fast process. All right, yeah, I'm going to move forward. So the last the last section is how we can actually use these retroviruses for good. And so as someone over there already brought up, there's this idea of gene therapy. And gene therapy is using viruses to fix genes that might be causing disorders. And so the idea is that genetic disorders are diseases caused by a gene that isn't working properly. Perhaps it's not producing a protein that you need for an important function in the body. And so the idea is that if we can use a retrovirus to put in a new copy, a working copy of the gene that's broken, then we can maybe fix the disease at the root of the problem rather than treating the symptoms. So just to clarify that a little bit, in many diseases, what we have is maybe, again, only a few bases, only a little bit of the DNA is off. But if that means that you can't produce a really important protein, that can cause a disease or a disorder that can be harmful. And one example that I'm going to use to explain this process is hereditary blindness. There are a few different types of hereditary blindness. So this is when the source of blindness is actually you've inherited it. There's a gene that was passed on that was not working or complete in some way. And we actually know for many of these different types exactly what the gene and what the mutation is that makes it not work. And for the particular type of hereditary blindness I'm going to talk about, there is a mutation in this RPE65 gene. It's a gene that has to do with vision, and if the gene is mutated, you can't absorb light properly, and therefore you can't see. So you have cases with babies being born blind or going blind at very young ages. So the idea of this therapy is to take a retrovirus. So, I mean, it could even be something like HIV or another retrovirus that knows how to integrate itself into the DNA of another organism. And we can cut out the middle, make sure it's not infectious anymore, and put in a new copy of this gene that's necessary. So we have here a new copy of this gene that doesn't have the mutation that might be causing problems. And then if we can inject this retrovirus into, in this example, the eye, and therefore have a new working gene inside, we'll now have the correct gene as well as the mutated gene, but now because the correct gene is there, we can produce the protein, this RPE65 protein that's necessary for vision. It's made correctly and actually restore vision. And this has actually been done in clinical trials over the last few years and been successful at curing very young children of blindness. So taking a step back, I think this is super crazy. Like, we can cut out viruses and put DNA into us. It's a crazy thought. And I just wanted to say that, yeah, we can definitely do that. I've actually used this technique in my lab, not for curing diseases, but for doing various types of lab work. And it's actually a relatively simple process because as I mentioned at the beginning, viruses are really simple. They're often just a few genes that are coded in a very small piece of DNA or RNA that make a virus. So it's actually quite easy to make a modified version of a virus in a lab and to put our own RNA into it. So if this is true, then why haven't we cured everything? Why is this not a miracle cure for all of these genetic disorders? Why haven't we cured, for example, colorblindness, cystic fibrosis? These are all disorders that we know are caused by a particular gene, and in many cases we know the gene, we know the specific mutation that causes them. Why haven't we fixed them? And the answer is that gene therapy is still very much in its infancy as a technology. Here I've listed just a few of the limitations. And some of these we can understand by thinking about the viral life cycle and some of the questions that we've discussed. So one problem that we're having is viruses can sometimes infect more than one kind of cell. They're not super targeted. And you can imagine that if you're inserting new DNA that produce new proteins in cells where they shouldn't be there, you could have some problems. So this is just one of the reasons why this retroviral gene therapy isn't perfect yet. But these are all problems we're working on and getting to good solutions for. Other ideas are that even though these viruses are not infectious, they're not going to make you sick, our body doesn't like foreign particles. Our immune system is pretty good at getting rid of them, and so if our body is not reacting well to this foreign particle, you might get another type of sickness as a result. And so this is just some of the reasons why gene therapy isn't perfect yet, but it's definitely in progress, and as I mentioned with the blindness example, it's actually been very successful in specific cases and has gone through clinical trials and actually helped people. And so with that, I'm going to wrap up this section of the talk. Just to summarize, what I hope you'll take away from this is that retroviruses can be really bad. They have this really unique way of getting into DNA and causing problems there. But they also can be sort of neutral. They can be the junk DNA that just sits on our genome and doesn't do anything. And the hope that is in the future and very soon, they can actually be used as a force for good. We can use these retroviruses and their ability to integrate to insert new genes and hopefully cure a lot of these genetic disorders. With that, I'll take my final round of questions before moving on to Katie's part. Yeah, in the front.
So the reason why they do it in really young kids is Is that why it works better in young children to do the?
So the question is why they do it in young children, as why they do that. That's actually a good question. I'm not sure if I know the answer. I'm not 100% sure that it has to do with the cells replicating. It might just be that they're prime candidates for this sort of study since they have their whole life of not seeing ahead of them. I don't know if anybody else has a better answer for that, but sorry. Other questions? Yeah.
Usually when you're doing experiments where you're trying to insert things into cells, you have to make some sort of attempt and then have a way of filtering or testing for which ones got what you wanted to get in there, which ones sort of missed or failed or got inserted wrong. I don't do it myself, so that's about my...
Yeah.
Anyway, so my question is, you must be trying to modify some template virus. You must be trying to modify it to put in the thing you want it to carry into your other targeted destination. How do you filter or test the viral particles? That you must be doing something, and then you must need to somehow filter the viral particles to figure out which ones are now carrying the thing you want or not. Now, the usual thing that I think about is that you're inserting something into an E. coli or something like that, and you put alongside the thing that you want carried in some sort of resistant, like
so that it can't.
Something that you can then use to test for. But the virus is carrying, you said, a really small amount of stuff. So what's your testing methodology?
Yeah, so just to simplify that question a little bit, the question is, how do you actually make sure that the viruses we're inserting have this like new DNA that we're putting in there, right? Like versus what they had. Yeah, so the sort of interesting about this is that when you do it in a lab, we often don't take like the whole virus and stick something in it. One of the most common methods is to take pieces of RNA that code for specific genes and put them in separately. So you put in like the envelope gene separately from the capsid gene, and you put them together in a way that the only way you can produce a whole viral particle is if the piece sort of gets part of it. So it's part of like fitting in the puzzle. You need like, let's say 3 pieces to create a virus, and one of the pieces has the DNA that you're trying to insert on it. And so if that piece doesn't fit in with the other 2, you just won't have a virus. So what you can do is you can mix these things together and test how many live viruses you have and be relatively certain that all of those viruses have it because it was attached to one of the pieces. Does that make sense? Yeah.
I was wondering, for gene therapy, how quickly is it effective? Like, does it gradually happen as more of the cells have the right DNA?
Yeah, so the question is, how quickly does gene therapy happen? And in most of the cases where we use gene therapy, you're putting in a gene that sort of is a replacement for a gene where like a protein wasn't being made. So there are all sorts of studies being done for like how much new DNA you need to insert to get enough protein that it starts working properly. So I think the answer to that question really depends on the gene, exactly what the protein does, like how the therapy is being developed.
I think we'll take one more question before the break.
Lucky last question.
So when you're doing these gene therapy testing, which retrovirus do you use?
So the question is, which retroviruses do we use in gene therapy? And that actually varies. One of the things that researchers are working out right now is which ones are the most safe and cause the least problems, and which ones can we target most specifically. Retroviruses tend to be quite simple though, so I think it's more of a matter of trying to find the right one for a particular gene and body part. But in the lab, for example, you use all sorts of retroviruses. Like you can use HIV because it works and you're not making anything infectious.
So my part of the lecture is about viruses and our bodies. So Shirlee just gave us a really great overview of how viruses can interact with our genome. I want to talk about the active viruses that actually, quote unquote, live inside of our bodies and provide us with some really unexpected benefits. And so the first part of my talk is going to focus on the human virome. I will define that for you in a second. And the second part of the talk we'll discuss a new piece of technology that allows us to really delve into our infection history. So let's start off with the human virome. And before I get into the really virus-specific part, I just want to introduce the concept of the human microbiome. And so remember from Shirlee's part of the talk that only 10% of the cells in our body are actually human. The rest are microbes, and this includes both viruses and bacteria. So the microbiome is the collection of microbes that live inside of our body and share our body space. And since the microbiome in our gut is the best characterized, that's the one I'm going to focus on. And the best studied members of the gut microbiome are definitely by far the bacteria. We have over 100 trillion bacteria in our gut alone. But we're now really starting to better understand some other members of our microbiome. And unsurprisingly, that's going to be the viruses. So a subset of the microbiome is the virome. And the virome is the collection of viruses that share our body space. And just to remind you of the scale we're dealing with here, each viral particle is about 200 times smaller than a human cell. But we still have a lot of these guys in our body. Just to give you a sense of the numbers, there are over 1 trillion viruses in just our gut. But in general, we haven't really heard as much about the human virome, especially compared to the bacterial microbiome. Why is this? And so the answer really lies in our tools to sequence DNA. So in our gut, we have human cells, we have bacterial cells, and we have viral cells. And so accordingly, we have human DNA, viral DNA, and bacterial DNA. When we want to find out the composition of the microbiome, viruses and bacteria included, what we have to do is take a sample of the feces from our gut, because that's going to contain all those cells and all that DNA, and try and extract the DNA from that. But this is where we run into a pretty big problem. So when we extract the DNA from our fecal sample, it turns out that when taken out of context, DNA is just DNA. No matter where it comes from, DNA is made up of the same code, the same 4 letters. So when we take DNA out of a virus and mix it into the same pile as DNA from humans and bacteria, we can't tell them apart right off the bat. So we have pretty good references for the human DNA and the bacterial DNA because we have the human genome sequenced, so we can use that as a reference for which of those sequences are human. And there are some really cool conserved sequences in bacteria that we can use to kind of map out what bacterial species might be in our gut. We don't have the same kind of references for the viral sequences though, and that becomes a huge problem. So one way I kind of like to think about this is, say you're posed with the problem of trying to sort these 3 types of sequences. This is kind of akin to being in a library and somebody's dumped a huge pile of books in front of you. Some of them are in Portuguese, some of them are in Italian, and some of them are in Spanish. Except someone else has decided to make your job really difficult and torn out all the pages in these books and torn out all the individual words and left you with an enormous pile of scraps. And they say, okay, sort them into Portuguese, Italian, and Spanish. So it's nice because you have some really, really awesome dictionaries for the Portuguese and the Italian, which in this scenario are the human and bacterial sequences. But for the viral sequences, the Spanish, you've got like a level 1 vocabulary list, and that's not going to get you very far. You've got a problem here because they're all using the same basic alphabet, and there are a lot of words that overlap between the 3 languages. So maybe you can sort out a lot of the Portuguese and the Italian, and maybe you can sort out some of the Spanish, but you're left with a lot of words that you don't know where to put. And so at the end of this, we always end up with this big junk category, the unknowns, the ones that we started with that we couldn't sort into one of our 3 main categories. And so this becomes a big problem because we still don't have a full understanding of all the viruses that live inside our body. If there are any new viral sequences in that mix, we can't tell them apart from something that's just unknown. So what's one way we can get around this problem? Remember that viral particles are much, much smaller than human cells or bacterial cells. And researchers have come up with a clever way to utilize this fact. Basically, all they have to do is filter out the viral particles, and all you're left with at the end of this size filtration are presumably viral particles that you can then directly sequence, and presumably at the end of this, all you have is viral sequence. And so even if you come across something that you have never seen before, you can more comfortably say, hey, that's got to be a new virus. Let's look into what that does. Okay, and I just want to pause here and take some questions on anything I've just said. Yes?
Using that method, how are you sure that you're filtering out any free DNA?
So that's a good question. I haven't actually personally done this, but the idea here is that we're using a filter just to filter out like the full particles, which is going to include the entire virus, the protein and the DNA inside of it. That's not going to go straight to like getting the DNA out. We'll have purification mechanisms to make sure we're like filtering out salt solutions, like whatever extra fecal matter was left in there, and other particles that are not viral. It's not perfect, but that really helps clear out some of the noise. That's a great question. Okay. Oh. Sorry, is there another question? Okay. All right, so I just talked about using some genetic tools to gain a better understanding of our virome, but why do we even care? Why do we want to know more about the viruses that live in our body? Can't we just say, cool, they're there, why does it matter if they're just kind of neutral? But it turns out we're starting to learn some really cool stuff about our virome, and certain members of it can actually protect us from disease. And now I'm going to take you through one of my favorite examples of this. So, in this scenario, you are kind of a mouse. The mouse is our model organism here, and it's our stand-in for a human because we would get in big trouble if we tried to do this with humans. So when you have a healthy mouse with a healthy, intact bacterial microbiome, when it encounters a pathogenic bacterium, in this case something called Citrobacter rodentium, which is kind of like the mouse version of really bad E. coli that you don't want to get, the mouse doesn't really care. The pathogenic bacterium will pass through its system, but there are enough good bacteria in its gut to kind of fight it off. And so at the end of this equation, we still have a healthy mouse. And it's all because it had a full intact bacterial microbiome. However, when we first treat this mouse with antibiotics, the antibiotics get rid of a lot of the good bacteria in its gut. And so the mouse no longer has a team of bacteria to kind of fight off this invader. So when the Citrobacter comes along, well, things don't end so well, and we end up with dead mouse. So researchers wanted to know if there was something else they could add to this equation to restore the healthy mouse at the end of this equation. What can kind of compensate for not having healthy bacteria in our gut to fight off bacterial invaders? And it shouldn't be shocking that the answer to this is a virus. So what happens here is, even though the mouse no longer has the full microbiome to protect it, adding this symbiotic virus that normally lives inside a healthy mouse but doesn't cause infection is enough to prevent other pathogenic bacteria from invading and causing disease. So in this case, a member of the virome is actually protecting its host. All right, and so with this example, I hope I've been able to show that there are some really cool parts of the virome and that they really are capable of protecting us. And so in this example, we have a virus that's mainly affecting host cells, the mouse cells. But this is actually a little bit of an anomaly, excuse me. It turns out that most of the members of our virome actually primarily interact with bacteria instead of host cells. And the type of the type of virus that I'm going to discuss next is called a bacteriophage, a type of virus that only infects bacteria and does not infect human or mouse cells. And these viruses and bacteria actually seem to coexist pretty symbiotically and pretty happily inside of our gut. So these bacteriophage, which I mentioned before, are again viruses that only infect bacteria. They have no effect on human cells, and usually they only infect one specific kind of bacteria. And these viral bacterial interactions play a big part in stabilizing how our gut microbiome, the bacterial part, really works. So let's go through an example of this. And I just want to remind you that this is not what happens in all scenarios, but I hope this was a good illustration of how viruses can affect bacterial populations. So we're starting here with just a normal population of bacterial cells, each of these here. And the circle in the middle is the bacterial DNA, which in bacteria is circular. And so some of these bacteria are infected by bacteriophage, or phage for short, and I'll be using the nickname phage throughout the rest of my talk. And so what's a little bit different than what Shirlee described in the first half of the talk is each phage will land on a bacterial cell and, like a syringe, inject its DNA into the bacteria. What happens next is sometimes when these phage do this, a piece of the phage DNA gets incorporated into the bacterial DNA, denoted by this kind of pinkish red here in the bacterial DNA. The rest of it is intact, but what's happening now is every time the bacterium produces RNA from its DNA and then protein from its RNA, it's also producing viral proteins. And in this scenario, this is not killing the bacterial cell. But what's important here is, so keep an eye on these cells with the red here. This can actually be really advantageous for the infected bacteria. They end up outcompeting the uninfected bacteria by a lot. And so this is what happened in a recent finding that came out of a group at Baylor. So there's a bacterium that lives in our gut symbiotically, it doesn't hurt us, called Enterococcus faecalis. And Enterococcus is capable of expressing phage genes. So after a phage has infected the Enterococcus, it carries the phage genes and is able to produce phage proteins. And when this happens, it can outcompete its neighbors. So if you take a look at the graph over here on the left, we see that the y-axis over here is a competition ratio. A competition ratio of 1 would be if the strains being compared were equally competitive with each other, and you start with a 50-50 ratio at the beginning and end with a 50-50 ratio. What's happening is, in the column on the left, these are the Enterococcus that carry phage competing with their neighbors, and they're outcompeting their neighbors by a lot, 1.5 versus 1. But the Enterococcus that don't carry the phage genes fail to outcompete their neighbors. So what's basically going on here is the Enterococcus, one species of bacteria, is using phage to get rid of other bacteria that they don't want around. So why can't we do this too? It turns out we can with something called phage therapy. And so remember that there are many, many, many kinds of bacteriophage, and each different kind is capable of infecting one very specific type of bacteria. And for a long time, we've been really interested in using phages to get rid of our unwanted bacteria. And this started quite a long time ago, in the early 1900s. So almost immediately after someone identified bacteriophage, and a cool piece of trivia here is bacteriophage is a combination of the words bacteria and phage, which means to eat. So these phages are not literally eating bacteria, but they are infecting them and causing them to die in a lot of cases. Almost immediately after they were identified, people were trying to use them to treat dysentery and trying to commercialize them for general use in the clinic. But right from the get-go, it shouldn't be surprising that people had a lot of misgivings about using phage therapy. People were introducing one infectious agent to clear out another, and people were really worried about the side effects. After all, these were still active viral particles, and even though we know today that they only infect bacteria, we don't know the full range of side effects that's possible. But really, the primary reason that phage therapy kind of fell out of fashion in the mid-1900s was the discovery of antibiotics in 1941. And this was kind of a huge, huge deal at the time. People thought this was a miracle drug. Infections that used to be fatal were suddenly curable by one visit to the doctor and one shot of penicillin. And people had thought they had just solved infectious disease. But even though this kind of spurred a shift away from phage therapy, almost immediately after people started using antibiotics, they also realized that when you start treating bacteria with these drugs, and you don't always do it consistently, the bacteria very easily develop resistance. And so the rise of antibiotic resistance made a lot of these drugs less and less useful. So people turned back to phage therapy and tried to make more out of that. And it was only 6 years ago that the first clinical trial in humans was performed and published. And what happened here was people were able to successfully use phage to treat antibiotic-resistant ear infections. So phage therapy is becoming more and more appealing in recent years, especially given the fact that antibiotic resistance is not going away anytime soon. And just to compare the two types of treatments, phage therapy is still effective against strains of bacteria that have become resistant to drugs. Also, importantly, phage therapy is very specific. If you remember that each kind of phage only infects one type of bacterium, it leaves other bacteria alone, and it leaves our human cells alone. And accordingly, no one has yet identified side effects, though again, I'll caution you that some may still be discovered. And it's very cost effective. Basically what I'm telling you here is we haven't entirely worked out all the kinks yet, and phage therapy is not something your doctor is immediately going to prescribe you the next time you go see him for a bacterial infection. But scientists are also trying to develop new drugs and small molecules to mimic the effects of phage without having to introduce active virus into the human body to kind of mitigate those potential side effects that we don't know about yet. So to sum up this part of the talk, there are a lot of beneficial things about the viruses in our bodies. Our virome is capable of interacting with and stabilizing our bacterial microbiome, and it can also directly benefit us as hosts, even protecting us from certain kinds of disease. And we're hoping that eventually we can really co-opt these viral functions in the form of phage therapy to treat infections broadly in the future. And I'm pausing again here to take any questions you might have. Yeah?
You said that viruses in our gut overwhelmingly interact with the bacteria rather than human cells. Is it more than 9-to-1, which is the human cells to non-human cells?
Yeah, so, okay, so the question was about the ratio of viruses to bacteria in our gut. Is it similar to the ratio of bacteria to human cells? Oh, no, I'm sorry.
I misstated the question a little bit. So you said that they interact, the viruses in our gut interact more with the bacteria in our gut than with human cells. Did I get that correct?
Yes.
Is it more than we would expect by just the population of human cells to bacteria?
Oh, I see. Okay, so yeah, so the question is more about, is the reason that viruses interact more with bacteria in our gut just a numbers game because there are so many more bacterial cells than human cells in the gut? Okay, so that's a really interesting question. I don't think that's quite the case because, again, it seems that most of the members of our virome are bacteriophage, which are just not capable of interacting with human cells. But it would be really interesting if like the ratio of bacterial cells to human cells in our gut was a driving force for like more phages to evolve in the gut. I don't actually know if that's true or not, but that would be really cool. Yeah.
You talked a couple times about viruses out-competing each other, like for example with the Citrobacter. What does that really mean for a virus that isn't fatal to the host? I mean, what does competition look like?
So, I'm sorry, I'm not sure I totally understand your question. So just to clarify, Citrobacter is a bacterium.
There's the presence of a virus. Well, what I'm trying to get my head around is if having one virus sometimes prevents having another. You use the term that the first outcompetes the second. But if the first isn't fatal to the cells, like what does it mean? How does that competition happen? It's not eating. It's like, what does it do?
So the question is about how viruses might outcompete each other, especially if they're not infecting and killing host cells. So the answer to this is pretty complex. I think there are a lot of different mechanisms that viruses use to outcompete each other. Sometimes, as in the case of the bacteriophage, they'll put some of their DNA into their bacterial hosts, and at certain times, those bacterial hosts will start producing so much viral protein that actually more phages will be produced at the expense of the bacterial cell. And so in that case, the more bacteria that are infected by that virus, they'll outcompete the others just by pure numbers. In terms of viruses directly interacting with each other, that doesn't seem quite as common because, again, viruses need to infect a host cell, so like a human cell or a bacterial cell, to actually replicate. So in terms of pure numbers, they'll need to go through another type of organism. Does that answer your question a little bit?
I'm sure it's very complex.
Okay, and if anyone has anything to add, please jump in.
I mean, evolutionarily, we really don't understand if two things are supposed to be symbiotic for the host. We aren't sure why one relationship doesn't favor the other, but maybe it's the balance that will help us evolve into something better. So it's something we're still trying to figure out.
And a couple parallels have been drawn between the balance of bacteria in our gut and the balance of viruses in our gut. So for instance, what we eat really affects the composition of bacterial species in our gut, and there's some evidence now that the case is similar with viruses, though it's probably through their bacterial host. So if virus A only infects this bacterium and there's a lot more of them, then of course there will be more virus A. Does that make sense? Yes?
What are the relationships between viruses and hormones? Like, you know, I had read in the 90s doing research with people with HIV and giving them growth hormone, and it would shift the disease into a diabetic condition versus an HIV. So it was again another swap. But does the hormones have any relationship with what happens with the virus?
I do not know the answer to that. So the question was about how the population of viruses in our body might be affected by our hormone balance. The only thing I know that has some relation to that is I know the hormones that do have some interaction with our gut, they can affect bacterial populations and thus affect the viral predators of those bacterial populations, but I'm sorry, I don't know beyond that. Yes?
My question is a little bit more about the gut microbiome as it pertains to bacteria than viruses, so apologies for that. But what's the mechanism, or the most likely mechanism for how the microbiome fights off infection? So in the example of the mouse, is it just competing for space, or is it some sort of other mechanism that makes it hard for Citrobacter to get into the mouse?
Yeah, great. So the question was about how the bacteria in our gut microbiome outcompete pathogenic invaders like the Citrobacter in the mouse example here. So that is an area of very, very active research. But what we do know is there are a lot of mechanisms. Sometimes it is purely just space. A lot of times it's nutrients. So if Citrobacter really wants to eat a specific thing, but there's only a couple that you swallowed, for instance, then the bacteria in your gut that are already consuming whatever the Citrobacter wants to eat, they'll really easily outcompete the Citrobacter. Another thing is sometimes bacteria will actively, just because there are so many of them in the gut, they'll start producing different kinds of metabolites that can actually fight off the Citrobacter. And of course, there's also the component of our immune system, which is also very active in the gut, and so that's used to the bacteria that live in our gut, so it kind of ignores them. But once something like Citrobacter comes along, that'll be recognized usually and driven out by immune mechanisms. But I'd love to talk to you more about this after the lecture because it goes on and on and on and on.
Also, if you're interested in the microbiome, we have a lecture from, I think a year and a half ago now, on the microbiome in our seminar archive. So you can check out our website. It has a lot of information there. Thanks.
Okay, let's take one more question. Yeah.
Right. So
does the symbiotic virus kill the pathogenic bacteria, or they just modify the bacteria to turn them non-pathogenic? And secondly, there was an early graph on which you show that the virus infected bacteria outcompetes the non-infected bacteria. How does that happen? Is it that the infected bacteria grows faster than the non-infected bacteria, or?
Okay, so there were two questions. The first one was, in this case with the symbiotic virus, how does that drive out the Citrobacter? Like, how is it functionally protecting the mouse host that lacks the microbiome? And so in this case, they actually are not entirely sure, but they have some hypotheses about how this might be working. One is that the virus might stimulate the immune system to be more active against the Citrobacter. Another is that the virus might stimulate some kind of interaction between the remaining members of the microbiome. So when antibiotics are given to this mouse, not every single bacterium in the gut is depleted. Some stay behind and may have some beneficial function.
It's not that the virus directly kills the
No, yeah. So this norovirus only affects the mouse cells, actually. It's not a bacteriophage. And the second question was about how the infected bacteria that carry the phage genes in our competition example outcompete their uninfected neighbors. And so they were also not entirely sure about how that's happening, but a couple possibilities would be that they are able to produce some viral proteins or even full phage particles. And oftentimes when a bacteriophage infects a virus and incorporates its DNA, that prevents other phage from infecting that cell and killing it. So it's like the bacteria that are infected with the virus have immunity against phage, whereas the uninfected ones don't. I realize it's a little confusing because they have already been infected, but just keep in mind that it's just the DNA that's conferring the immunity and not like a living virus that's killing the cell. Does that answer your question?
No. That's
okay. Feel free to come talk to me after the lecture. Okay. Okay. And for the last part of my lecture, I want to tell you all about a new piece of technology that actually just came out of Harvard Medical School. It's called VirScan, and it's a way for us to uncover every infection our body has ever had. Okay, so we know that viruses are part of our genome because of retroviruses. But there are other ways that our body can actually store viral information, including the viruses that have infected us in the past and maybe caused disease or just passed through our body. But before I go on, I want to ask everyone in this room, is there anyone here who can remember every infection you've ever had? Every single one. Raise your hand. Stand up. Yell it out. Bring it on. No? Okay. Yeah, me neither. But luckily, our brain isn't the only part of our body that has memory. We also have an immune system that is fully capable of remembering all the microbes that have ever infected us, even the ones that didn't cause disease or the ones that are still waiting inside our body and may yet still cause disease. And so I want to focus today on a very specific part of the immune system and introduce the concept of antibodies. So this over here, this Y-shaped thing, is an antibody. And this is a protein that's produced by immune cells called B cells. And what antibodies do is they bind things called antigens. And antigens are just pieces of things that could do damage to our body. So these can be anything from pieces of bacteria and viruses to dust particles to even parts of damaged human cells. Basically anything that our body encounters and could affect our body negatively. So in a way, antibodies are kind of like bouncers or a police force that stops everything they encounter and asks them to show ID. If the ID that's shown is something bad, they will bind the antigen and recruit other immune cells to come and destroy it. And importantly, once an antibody encounters one of these antigens once, those B cells that were making the antibodies will actually produce a subset of B cells called memory B cells. And these, as the name implies, will remember this interaction. And so they'll wait dormant in the body even after the infection has passed. And so if our body ever comes into contact with that antigen again, those memory B cells are lying in wait to produce a lot more antibodies so our body can fight off that infection faster and more effectively. So it's like this police force keeping mugshots of these antigens so that if they ever run into them again, they'll know right away what to do. So our antibodies are kind of an effective archive of every infection we've ever had because of these memory cells. And this kind of information can become very important when we want to find out if we're infected with something. So if my doctor wants to find out if I have hepatitis C, they'll sit me down and take a blood sample. And what happens is they will have a known hepatitis C antigen that they'll test with my blood. But it's only an antigen for hepatitis C. But they'll mix that antigen with my blood, which contains lots of different antibodies. And if there are antibodies in my blood that recognize this hepatitis C antigen, so that means they're hepatitis C specific antibodies, they'll bind. And this test allows us to pull antigen bound to antibody out of the rest of the solution. And if that's the case, then it's a positive result. And yes, I do have hepatitis C. But if it's a negative result and nothing bound to the antigen that they mixed with my blood, we're back at square one. We don't know anything except that it's not hepatitis C. So it would be a lot easier if we could test all of our antibodies at the same time. If we have a mystery infection, it's much easier to ask all the questions at once than come up with one hypothesis disease at a time. And this is the idea behind VirScan. So VirScan, instead of just testing one antigen at a time to look for one individual type of antibody in our blood, they have a whole library of antigens. But remember that antigens are proteins, and proteins are not very stable. They're kind of hard to store long term. So the researchers came up with a really clever way to kind of store this information in the long run. What they do is they have this whole library, antigens from over 200 species of viruses, all the viruses that are known to infect humans, and they have over 93,000 types of antigens that come from different parts of these viruses. And what they do is they code for these different antigens with different parts of DNA. And as a vehicle to carry the antigen and test it with our different antibodies, they'll put this DNA into our familiar friend, the bacteriophage. So basically what's happening here is instead of the bacteriophage presenting their antigen to our immune cells, they're kind of stealing the IDs of all of these viruses over here, all 206 species, and carrying those IDs instead to show our antibodies. So that when these bacteriophage are expressing things on their surface, indicated by, for example, this blue thing here or this green thing here, these are not inherent to the bacteriophage. Instead, they're the IDs of other viruses that we want to test. And so we mix these bacteriophage, again, with the same kind of blood sample, which contains all of our different antibodies. Some of them will bind. So if we have been infected with whatever normally carries this blue thing, some antibody will bind. Same thing with this red one over here. And the bacteriophage expressing antigen that does not bind our antibody gets washed away. And so we're left with only bacteriophage expressing antigen that binds some of our antibodies. Then we can sequence these bacteriophage, which are carrying DNA coding for these specific antigens that our body recognizes, and that gives us a short list of all the viral infections we've had. And so when the researchers did this, they tested over 500 people from all around the globe, and they found that each person had about 10 previous viral infections, though some had up to 84. So let's compare VirScan to our current tests, and our current test is the hepatitis C example I used a couple slides ago. So compared to our current tests, VirScan can detect any virus known to infect humans with one test. Instead of going one at a time, hepatitis C or HIV, we're getting all of them in one go. It's also fast. It only takes a couple days, just like our current tests. It's pretty high accuracy, and importantly, it can identify asymptomatic infections. So even if you're infected with something, but you're not actively sick, a test with VirScan is a catch-all. Your antibodies will have noticed it even if you haven't physically noticed it. But something to keep in mind here is that false positives are possible in both tests. And by a false positive, I mean accidentally saying your body has come into contact with mono before when it actually hasn't. So VirScan hasn't actually been commercialized yet. This is not something we're actively using in the clinic, but it's pretty cheap, very effective, and the researchers are really hoping that they'll be able to do this soon. So in this last part of the talk, we've discussed how antibodies help maintain our immunological memory. We now have some really great tools to find out what viral information our body is carrying. And with tests like VirScan, we might be able to get around the burden of testing for different viral infections one pathogen at a time. So let's come back to this list of mainly negative terms that we saw at the beginning of the talk. I really hope that after our lecture today, we can start introducing some positive associations as well, and that you've learned a little bit of something. And I just want to conclude by saying that we've really just barely scratched the surface when it comes to learning about viruses. There's still a lot left to discover, both good and bad. And the truth of the matter is that viruses will probably still continue to cause disease in humans for as long as we're around. It's what they have to do to survive, but we're hoping that we'll come to understand them in a way so that we can use them to combat disease as well. And with that, I'll take any final questions. Yes?
Can you give an example in which VirScan would be used? Like, I'm just trying to think of why someone would go and get a scan. If they may have an undetectable virus that can be detected, but how would they know to go and get a scan?
So that's a great question. I think that becomes especially relevant when you have really dangerous infections that don't tend to show symptoms for a really long time. So like an HIV test or a hepatitis C test. So those are two examples of viral infections where by the time you start to feel symptoms, it's maybe too late. But it's not like if your hand turns blue, it's time for a VirScan test. Yeah. Yes?
So one thing about the VirScan, and I'll explain this if people don't know, with antibodies that are created by your body in response to infection, it's the same mechanism that vaccines work with. So vaccine has a live and weakened version of a virus, or a dead one, and so it has an antigen, and then your body recognizes it, produces antibodies. So I'm wondering, can the VirScan distinguish between like the presence of an antigen or antibodies from a vaccine versus from a virus? Because like I have a vaccine, but I know I definitely don't have or haven't had hepatitis A. Would it be able to tell the difference?
Yeah, that's a great question, and I think that is where some of the false positives come from. And so, like, but that is actually a problem with several tests that we have. So, for example, one of the tests for tuberculosis is like a skin test, and they'll inject some antigen into your skin, and if it swells up, it means your immune system's reacting. But for example, if some of you may have had the BCG vaccine, you'll also react even though you don't have active TB.
Or if you get that test too many times, it'll react.
Yeah. Yeah, so I mean, there are definitely false positives, and that's a huge problem with any kind of immune test. But yeah, that's a great point. Other questions? Yeah.
Similar to follow up with that question. So I think traditional diagnostic was you throw in a cocktail of antigens, hoping that your antibodies will recognize, presumably hep C. So in this case, how many of these bacteriophages are expressing different the epitopes for hep C?
Okay, yeah, so the question was about, so traditionally we want to cover all our bases. So when we test for an antigen-antibody interaction, we don't just want to put in one antigen for hepatitis C and hope that that's the right one. We want to put in a bunch of different ones just to make sure like our body is recognizing that set of antigens. And so with VirScan, they actually, so they're testing about 200 strains of viruses, but they have over 93,000 different antigens. So they are really covering their bases. Yes?
Well, my question is related to the vaccine question, and it is whether the typical vaccine, if I could get a vaccine for chickenpox, how many of the signature proteins on the real chickenpox is the vaccine duplicating? So would it be the case that my immune response on, like if you've got 1,000 different viral strains and VirScan is doing 93,000, so that's about an average of 90 per virus. So let's say chickenpox has 90 interesting things about it that my body could recognize if I had the real chickenpox. But does the vaccine only stimulate my body with 40 of the 90? So if I've had the chickenpox vaccine, am I going to get 40 positives on my VirScan, whereas if I had the real chickenpox, I would get the full 93? So my question is, do you know whether vaccines are providing, what fraction of coverage are they? So could we distinguish the results in the VirScan?
Okay, I will try to repeat your question accurately. Please correct me if I don't do it correctly. So this is related to our earlier question on vaccines. So vaccines work in a similar way in that they are trying to prime our body with antigen so that those memory B cells will be made and we'll be able to react when the real infection comes. And I believe the question was about how many different types of antigen are in vaccine formulations such that we can distinguish between someone who's just been exposed to a vaccine and only experienced like 4 types of antigen from chickenpox versus like all 100 that might actually occur if we got real chickenpox. And after that long-winded thing, I'm going to say I don't know. But does anyone else know?
It's not.
Yeah,
Okay.
variable either way.
It depends on the host response and like how you design vaccines. So
Yeah.
certain vaccines have like live viruses and certain vaccines have attenuated viruses, and we think that the immune response to each of those is different and will potentially generate different antibodies. But which of those are actually totally relevant for depending on the infection is also debated.
Yeah. And I also know that certain antigens, even if they're all in the same virus, some are more potent than others, so some are capable of stimulating the immune system more than others. And as Vinny just mentioned, we don't always have a good sense of which ones those are. Yes?
200 some odd viruses known to affect the human body seems really low, but is it that compared to the number of bacteria that infect us? It seems low. I mean, does it to you?
Yeah, okay, so the question was, is it really only 200 some types of viruses that infect us? That number seems low, and I totally agree. So kind of the subtext there is it's 206 dedicated species of bacteria. It's over 1,000 strains that they're testing in VirScan. And also, we have definitely not identified every virus that is capable of infecting humans, but I also will say that given the like 10 to the 32nd power number of viruses that are on this planet, most of them don't actually care that much about humans, so they'd much rather play with their bacterial friends. Yes?
So somebody had asked about a clinical application for VirScan, and I work in oncology, and I know that for certain treatments that we give patients, they become profoundly immunocompromised, and we definitely test them for all the viruses that we know of, so that we can treat them prophylactically viral infections that could occur when they were immunocompromised. So there's one application.
Yeah, that's awesome. And I know that, the question / comment made here was that there's potential for like cancer immunotherapy with VirScan, and that's actually something that they're really excited about. So in certain types of cancer, the body can also recognize cancer antigens, and so there's potential for immunotherapy with things like VirScan as well. Yeah?
So the 206 viruses that affect humans, those are viruses that affect human cells, right?
Yes.
And there's some other large number that affect bacteria cells that are commonly found in humans.
Yes. Yeah.
Which
So
are those? For this particular, that's part of the VirScan system.
Yeah, so the clarification there was that the 206 viruses mentioned in the VirScan context are those that directly affect human cells, infect, and cause disease in human cells. We are not counting the vast number of viruses that affect the bacteria inside of our body and have indirect effects on our health. Great, thank you so much.